Good morning, students. Today we are going to learn about processing of pulses, in which we are going to learn the effect of soaking, decortication, germination, fermentation, and cooking on pulses, along with the factors which affects the cooking quality of pulses. So pulses. So pulses are dry edible seeds of legumes. Example: lentils, dry beans, broad beans, field peas, lupins, chickpeas, bambara beans, pigeon peas, wedges, and cowpeas. It contains protein, dietary fiber, resistant, slowly digestible starch, polyphenols, saponins, lectins, phytic acid, and enzyme inhibitors. Storage proteins occur mainly in the cotyledons, and protein and starch are embedded in the cellular matrix with the seed coat acting as a protective barrier. Whole uncooked legumes are hard and difficult to masticate. The protective cell wall and the protein starch interaction restrict proteolytic enzyme access. Processing is therefore required to improve the edibility, digestibility, and sensory quality of pulses. The processing principle, which includes the objective, which includes the reducing cooking time and energy requirement, soften the seed and obtain acceptable texture, increase protein and starch digestibility, reduce anti-nutritional factors, increase the bioavailability of the minerals, reduce weeny flavor, and inactivate the microorganisms and enzymes. The sequence involves in processing as dry pulse, which is cleaned, sorted, and then get into the soaking procedure, optional decortication for some legumes, and optional germination or fermentation according to the need, cooking, and ready to eat product. Soaking will acts via the mass transfer. Dehulling acts as a physical dehulling or decortication via the physical removal. Germination and fermentation, which includes the biochemical reaction, and thermal treatment softens the seed and gelatinizes the starch. The soaking will affect the uh, cooking time, and also it will soften the pulses. Lentils and peas can be often be cooked without prior soaking. The mechanism involved in soaking is hydration, which involves the uptake of water and swelling of the seed matrix, followed by mass transfer. The soluble compounds will migrate into the soaking medium. Then the enzyme will be activated. This hydration will activate the endogenous enzymes such as phytase, and the softening of structure will happen, and it will respond faster to the heat. The phytate reduction is linked to the endogenous phytase activation and diffusion of breakdown products into the soaked water. Overnight soaking followed by discarding the soaked water can reduce the oligosaccharides by up to 75%. The effect on nutritional composition, it will reduces the phytate, tannins, trypsin inhibitors, oligosaccharides, saponins, and some polyphenolic compounds. It can enhance the protein and micronutrient bioavailability, lower the antioxidant and prebiotic potential. Some beneficial bioactive compounds are also leached out. Effectiveness varies with the pulse type, soaking conditions, and the chemical structure of the target antinutrient. Soaking is a hydration or a mass transfer balance which with reduced anti-nutrients and cooking time, and also the leaching of water soluble nutrients also takes place. So, as per the research findings, when the pulses like green gram, horse gram, lentil, chickpea, cowpea, which was soaked for 16 hours in water, will increases the protein digestibility for about 2 to 5%. The bambara groundnut, which is soaked for 48 hours with in the temperature of 30 degrees Celsius in 1:10 water ratio, the tannin has been decreased about 99 to 100%. Phytic acid also decreased by 55 to 68%. Trypsin inhibitor will decreases for about 33 to 43%, and digestibility has been increased by 9 to 11%. Grass pea, which was soaked for 9 hours and an ambient room temperature, will reduces the tannin content for about 63%, phytic acid content for about 62%, and digestibility has been increased for about 1%. And kidney beans, which was soaked for 16 hours in an ambient temperature, will result in the reduced tannin for about 21 to 31%, and phytic acid also has been reduced to 13 to 19%. Effect of decortication or dehulling in the pulses. So dehulling is nothing but the physical removal of the outer seed coat and exposing the cotyledon and reducing the barrier to hydration and heat penetration. It can improve the protein digestibility and flour functionality, and also improves the visual quality. The faster cooking can be attained when the seed coat is removed, and it will reduces the phenolic compound content, and removed hull can be used as a fiber byproduct. The research findings about the dehulling black bean, when it was dehulled, the trypsin inhibitor will be reduced to 17%, and the digestibility has been increased from 65 to 70%. That is increase in 5% of digestibility. Bambara groundnut, when it was pre-soaked and dehulled, the phytic acid has been reduced by 3% in one variety and 5% in another. And the velvet bean, after dehulling, the digestibility has been increased by 3%, and dehulling plus soaking has increased the digestibility for about 82%. The germination, it is an inexpensive bioprocess in which the dry seeds absorb moisture, break the dormancy, and the embryonic axis elongates as the metabolism resumes. The water uptake activates endogenous enzymes, and the storage compounds mobilize to support the embryo growth, and the anti-nutrients are degraded or transformed, and the new metabolites may form, and it has a good effect on the nutritional availability. The nutritional effects will be the anti-nutritional factors has been reduced, and it will increases the beanie taste, and improves the protein and starch digestibility, and improves the amino acid content and trace mineral bioavailability. It can increase total dietary fiber and soluble sugars. Protein and fiber concentration may increase due to the biochemical redistribution and dry matter changes. So the important parameters which should be considered for germination: the temperature, water availability and the quantity of water, oxygen, light, duration, and the type of the pulse seed, hygienic and environmental condition. And the outcomes of the germination will be strongly dependent upon the variety of pulse, and also the digestibility of the grains under different temperature and duration combinations. So the germination, the black bean, when it was soaked for 24 hours in a 20 degree soaked and then germinated for 24 hours in 20 degrees Celsius, the digestibility has been increased about 9%. PG&P for 48 hours at 25 degrees Celsius, the germination will took place, and the digestibility has been increased to 13%. And for lentils, the germination period is 48 hours at the temperature of 25 degrees Celsius, and the tannin has been reduced, and the phytic acid also reduced, and the digestibility has been increased from 77 to 78%. And for grass pea, the germination time is 48 hours at the ambient temperature. The tannin has been reduced to 80, for about 86%, and phytic acid also reduced to 91%, and the digestibility has been increased by 5%, with 75% to 80%. And the fermentation, so the biochemical processing method and the microorganism and their enzyme transform the pulse component and may be natural or controlled solid state, may occur in solid state or in liquid state. The key parameters for fermentation will be the microbial culture, enzyme activity, fermentation method, and the environmental condition. In this, the mechanism involved, the microbial enzymes will hydrolyze the complex component, the phytate and the anti-nutrients are degraded, and the protein structure is modified, and the organic acids improves the mineral solubility, and the oligosaccharides are metabolites, and the flavor aroma compounds has been generated. The natural fermentation can partially or completely remove the alpha galactosides, tannins, phytic acid, lectins, and trypsin inhibitor activity. The fermentation, the lentil, Pleurotus austriatus, for 28 degrees Celsius, at 28 degrees Celsius for 14 days, the digestibility has been increased by 15%. And for Zamni, the pre-boiling plus the involvement of rhizobus or azae at 30 degrees Celsius for 48 hours, the phytic has been reduced below the detection mode. And for grass pea, along with Lactobacillus plantarum at 30 degrees Celsius for 48 hours, the phytic acid has been reduced, and the tannin has been reduced. The digestibility has been increased about 6%, and the faba bean flour along with Bacillus pumilus at 37 degrees Celsius for 72 hours increases the digestibility for about 78% and decreases the phytic acid content below 9%. And effect of cooking and thermal processing. The cooking is most important because it is the uncooked legumes remain difficult to masticate. The steam, by using steam or using boiling water, you can cook the pulses, so it will give you a palatable texture. So the high temperature will enhance the thermal solubilization of pectin in the cotyledon cell walls. Cell wall barrier is disturbed and the improves the protein accessibility. Starch gelatinization increases the susceptibility to enzymatic digestion. Protein denaturation improves the accessibility to the digestive enzymes. Heat reduces or inactivates heat-sensitive anti-nutritional factors. Cooking also inactivates enzymes and microorganisms, contributing to product safety and stability. So there are different types of cooking which involves boiling and pressure cooking. For boiling, which is the most common thermal method, the LOP for 30 minutes boiling has been reduced the tannin content to 46%, and the digestibility has been increased to 92%. And for grass feed, the 3 hours of boiling will reduce the tannin for about 27% and phytic acid to 34%, and the digestibility has been increased to 79%. Pressure cooking, which is more intense than boiling, which permits the shorter processing time and usually 15 to 20 minutes. This also shows that the trypsin and phytic acid and tannin contents has been reduced and the digestibility has been increased by 10 to 15%. So other thermal methods of cooking is roasting or dry heating, microwave cooking, autoclaving or steam sterilization, and infrared heating or microionization. So for roasting, there is no need of water. It will improve the shelf life, flavor and aroma. It can reduce the anti-nutrients and improve the digestibility. For microwave cooking, it uses water but a very short time. It can retain the sensory quality and protein structure becomes more disorder. And trypsin inhibitor has been reduced to 93 to 0.29% and tannin 34.38% and phytic acid 39.17%. So the different processing methods and the benefits will be the soaking. The main mechanism is hydration plus mass transfer. It will shorten the cooking time, lower the anti-nutrients and better bioavailability. For dehulling, the seed coat has been removed and the better access and functionality has been attained and lowers the phenolic content. And the germination, the biochemical activation took place, which lowers the anti-nutrients, better digestibility of amino acids and minerals. And also fermentation, there will be a microbial transformation took place, lowers the anti-nutrients, improves digestibility and palatability of pulses. Boiling, hydrothermal treatment as a mechanism, and the soft texture, start gelatinization and protein denaturation takes place. In pressure cooking, high temperature and pressure will increases the rapid and strong digestibility improvement. In roasting, that is in the dry heat, the flavor and shelf life has been increased and the anti-nutrient has been reduced. In microwave and infrared, the rapid electromagnetic heating will reduces the processing time and the anti-nutrient has been reduced. So factors affecting the cooking quality. The cooking quality is nothing but the ability to hydrate, soften and reach the acceptable texture in a reasonable time while retaining appearance, flavor and also the nutritional quantity. So the pulses, the type of species of pulses and the seed coat structure and cotyledon composition will differ according to the species. So that will affect the cooking quality of pulses. Seed size and shape, which will influence the hydration and heat penetration of pulses. Seed coat thickness and the permeability will affect the water uptake by the pulses, and the cotyledon composition, that amount of starch, protein, fiber and pectic substances present in the cotyledon will affect the cooking quality of pulses. The storage duration, the how long we are storing, that based upon the storage duration, the cooking quality of the pulses has been reduced. And then the temperature and humidity where it was stored, the seed damage, seed coat darkening can promote the hard to cook behavior. So the soaking factors like duration, water to seed ratio, the amount of water we are adding, and then temperature, the pH we are maintaining, the hardness of the water, the type of the water we are using for soaking, and the soaking medium, and the discarding or retaining of soak water, and the uniformity of hydration within the pulse seeds. The cooking medium factors like water quality and minerals, pH, and salt, mineral ions, and water volume. Cooking process factors like temperature, time, pressure, heating rate, moisture content, pre-soaking degree of dehulling and milling, and combination with the germination or fermentation. So when it was germinated and or fermented, it will has less time to cook. And milling increases expose the surface area and breaks cell walls and reduces the cooking time and increases the nutrient availability. Infrared treatment can modify pectin and shorten the cooking time, though excess treatment darkens the color and hardens the texture. Hard to cook phenomenon, pulses that require unusually long cooking and fail to soften adequately, it is an important quality defect. So the general causes is unfavorable storage, changes in seed coat permeability, cotyledon and cell wall changes, pectin mineral interaction, and inadequate or non-uniform hydration, and also due to genetic or cultivar differences. So this will make a longer cooking time, higher energy use, poor eating quality, reduced consumer acceptance, and possible nutrient losses when it was cooked for a long time, and non-uniform texture. So we can use sound and properly stored pulses. Adequate pre-soaking is necessary. Pressure cooking if it is in hard seal. And then dehulling or milling, optimized cooking water chemistry and pH to be maintained, and controlled pre-treatments can be done. And combined processing like germination, soaking and germination and fermentation, rather than excessive severity. So these are some possible sequence and expected outcome, like how if you are making an household curry, you can soak, boil or pressure cook so that you can reduce the cooking time, get a softer texture and better digestibility. If you are dal you are going to make, you can decorticate, split and then cook so that the rapid hydration and soft cooking will be attained. For fermented pulse food, you can soak and then ferment and then cook or dry. That will improve the digestibility. So the soaking is the hydration and mass transfer, which will reduces the cooking time and several anti-nutritional factors. So these are the summary of dehulling, germination, fermentation, cooking, and hard to cook defect. So the pulse processing responses are pulse and condition dependent, and the parameters must be tailored to pulse and the product. Thank you. Happy learning.